Good morning or good afternoon, dear attendees of the Discovery User Symposium 2020. This is Dr. Kombak from Precision Pathology Biobanking Center of Memorial Sloan Kettering Cancer Center in New York. Our PPBC is the core facility for research operations in MSKCC, consisting of histology and immunohistochemistry labs and the biobank. I myself, I'm a pathologist. I did my medical training and residency in Turkey, in Istanbul, and I've been working in this facility for almost 15 months now, under supervision of Dr. Roll, Dr. Silber, and Dr. Benot, and with a great team, of course. In this presentation, I will try to demonstrate our multiplex immunohistochemistry experience with you, immunohistochemistry experience using the Discovery Ultra platform. First of all, I would like to quickly revisit the basics first. Immunohistochemistry is a widespread technique which we use in our daily practices for tumor diagnosis and classification, prognosis and prediction, and also for selecting candidates for targeted therapies. With IHC, we can selectively identify antigens with specifically binding antibodies and visualize antigen-antibody interaction with detection systems. There are a lot of detection systems which are direct conjugate labeled antibody method, indirect or sandwich procedure, unlabeled antibody methods, White-in streptavidin systems, and alkaline phosphatase labels and polyvalent detection systems. The most common ones we use are the peroxidase, antiperoxidase method and the AP anti-AP method from all of these. We also need chromogens to visualize the end product. A chromogen is a color-producing substrate which is activated with the enzymatic reaction of the detection system. Chromogens are either peroxidase-based or alkaline phosphatase or glucose oxidase-based. The most common ones are DAB, AEC, and FastRat we usually use in our daily routine. New chromogens are also emerging, such as horseradish peroxidase-based ones, which I will talk in detail later. Here's a simple demonstration of most common chromogens. On the left-hand side, you'll see the red membranous staining of AEC, and on the right side, you'll see the brown membranous staining of DAB. There's been a growing demand about immunohistochemistry, like if only we could label more than one marker per section, or assess combined markers like PD-1 and PD-L1, or be able to see the distribution of immune cells and interaction between cells and or molecules, and in the meanwhile to preserve tissue and time. These ideas led us to the concept of multiplex immunohistochemistry. Today we have a bunch of different techniques for multiplex immunohistochemistry procedures, which are Brightfield-Based, metal-based, chip cytometry, or DNA barcoding-based methods. Today I'll be talking about the Discovery Ultra Staining System, which is an example of Brightfield-based multiple immunohistochemistry. It is an automated platform for research purposes, and it works with sequential cycles without these staining steps in between. It uses chromogens with broad absorbance spectra and doesn't need an integrated scanner or imaging microscopy or analytic software. It is easy to perform and easy to evaluate. You only need a conventional bright field microscope to evaluate the end product. On Discovery Ultra Staining System, you can combine AP and HRP based chromogens in sequential cycles. So to explain that, after deparaffinization and pretreatment, there are consecutive cycles and antibody denaturation steps in between. Each sequence consists of primary antibody, secondary antibody, enzyme conjugate, and chromogen incubation periods. And after the last cycle, there is a step for counterstaining with hematoxylin. Our 5-plex design consists of PD-L1, PD-1, CD68, CD8, and CD3 antibodies. And we combined AP-based red and yellow stains with HRP-based DAB purple and teal. PD-L1 is the programmed death ligand 1, which is a transmembrane protein that has been speculated to play a major role in suppressing the adaptive part of immune system. That means the upregulation of PD-L1 may allow cancers to evade the host immune system. Many PD-L1 inhibitors are in development as immuno-oncology treatments and are showing good results in clinical trials. So PD-L1 assessment has been a center of interest for a while. Just to show simple examples for antibody chromogen couples in our design, you see here the brown membranous staining of the DAB for PD-L1. Purple for PD-1. There are some teal staining and CD8 positive cells around, but the purple staining is still distinguishable. Red for CD68 to highlight macrophages. Teal for CD3 and yellow for CD8. These two are usually intermingled in our samples. We applied this 5-plex protocol on our collection of pan-tumor tissue microarrays. These pan-tumor TMAs are three separate blocks with a total of 144 cases, which are carcinomas of different origins like lung, colon, stomach, and gallbladder and pancreas, as well as gastrointestinal stromal tumors, sex cord stromal tumors, and soft tissue and bone tumors, melanomas, and other rare and miscellaneous tumors. This application allowed us to have a broad opinion on the distribution of PD-L1 and PD-1 expression and the immune response in different types of tumors. Here is an example of a PD-L1 positive core. It is a nice display of the tumor and the strong PD-L1 positivity and the polyphonic immune response around it and also within the tumor. If we look closer, we can get a better detail of the PD-L1 expression in the tumor and also the immunoprofile of the infiltrating cells in the scene. In some of the cores, we observed some focal mild red staining, mainly on tumor cells, which seemed like a precipitation issue and improved with an additional step of reaction buffer rinse. And after that addition, I can say that the background staining is minimal, as you can see here, even in areas with dense immunoinfiltrate with a lot of positive cells. Another material we run the procedure is the pancreatic ductal adenocarcinoma tissue microarrays, which are 2 separate TMA blocks carrying a total of 87 cases, 45 of which are female patients and the rest of 42 are male patients. Also, 36 of them are short-term survivors and the rest are long-term survivors. And the median age is 70. I displayed this image to give some opinion about how I interpreted these TMA sections with the 5-plex. On the left-hand side, you will see the green annotations for tumor areas, and on the right side, you will see the pink annotations for peritumoral stroma. As is known, the tumor and stroma can be very intermingled in PDACs, and they both contribute to the malignant process, but still this discrimination provided me so much information about the tumor-associated immune response. Here is an example of a PDAC case with some degree of PD-L1 expression, and also we can see the infiltrating immune cells with different immunoprofile. You can also measure the proximity of PD-1 positive cells to the nearest tumor cell with using a simple image analysis program, or it can be a freeware program online. I would also like to talk about another design we created using the new chromogens, which are the beta products of Roche Tissue Diagnostics, and they're not commercially available yet, and they're still in development. And they are tyramine-based HRP-mediated chromogens. The procedure is sequential, just like the 5-plex design, this time only with the HRP-based chromogens. So similarly, after deparaffинization and pretreatment, there are 3 consecutive cycles consisting of primary antibody, secondary antibody, enzyme conjugate, and chromogen incubation. And in the end, there is a step for counterstain with hematoxylin. In this design, we combined PD-L1 and TTF1, PD-L1 matching with red chromogen and TTF1 matching with the yellow chromogen. Both are HRP-based. And we used 6 pulmonary adenocarcinoma cases with varying levels of PD-L1 expression. Here is the yellow nuclear staining for TTF1 in tumor cells, and the red membranous staining for PD-L1, and the combination you see here. You see here the PD-L1 positive and TTF1 highlighted tumor cells in the upper half. There are also some other red staining cells beneath that part, but most of them are macrophages. Actually, it is hard to discriminate on monostain whether the PD-L1 positive cell is really a tumor cell or a macrophage, especially in tumors with solid pattern like this and abundant macrophages around. They can be positive for PD-L1 and easily be misinterpreted as a tumor cell. So marking tumor cells with TTF1 at the same time facilitates the interpretation and therefore provides a better PD-L1 scoring. Similar to this image in this one, there is an area of macrophages in the center of the image, which can easily be identified as PD-L1 positive tumor cells on monostain. And this is another case showing a smaller percentage of PD-L1 positivity. And as you can see, the staining intensity is variable from weak to strong in this sample. As an adjunct, we decided to add cytokeratin as a third antibody in the design. So it would cross-check and enhance the efficacy of TTF1 to detect the tumor cells, creating a discernible transition from nuclear yellow staining to the membranous red staining. We used the same 6 lung cancer cases again for this design. And you see here a part of benign epithelium staining with blue for cytokeratin and yellow for TTF1. This is the case with lower PD-L1 positivity I showed you earlier with the double design. Now the tumor cells are highlighted both with yellow staining of TTF1 and blue cytoplasmic staining of cytokeratin. Blue cytoplasmic staining of the cytokeratin makes it easier to detect the red membranous staining of PD-L1. Now let's review some comparison images with DAB. This is a case with negative for PD-L1. There is some mild stromal background staining with DAB, which we don't see in the triplex design. And in this sample, it's a case with mild to moderate positivity for PD-L1. And I tried to capture the exact same areas to compare the staining quality. I can say that the variation in staining intensity is with the red chromogen is very similar to DAB, and the percentage of positivity is also accurate. Another case with moderate staining. Red staining is moderate in intensity likewise. The same subpopulation, however, on the left side seems somewhat paler in both stains. This is partially because of the infiltrating lymphocytes, which can easily be recognized and ignored with TTF1 and cytokeratin negativity in the triplex design. and an example of strong positivity. When the membranous staining is strong, we usually get some cytoplasmic reflection of it with any chromogen. Blue cytoplasmic staining of cytokeratin also helps with that by overruling that infusion-like staining from the membrane. Now a closer look in 4DX to the same area. The red membranous staining is very sharp and strong, just like DAB, which gives me the idea that it can be implemented to HER2 staining also. And also a lot of other combinations can be created with other antibodies. So based on our experience, I can say that Discovery Ultrastaining System is an efficient automated platform for research purposes. On this platform, you can combine AP and HRP-based chromogens, and it's very easy to perform. With this stainer, you don't need to integrate a scanner or an imaging microscopy or analytic software, and it's easy to review. You only need a conventional bright field microscope, and you see all the chromogens on the same slide under light microscope. There may be minimal overlapping and minimal background staining due to the utility of multiple chromogens, but if you have such a problem, I can suggest to add a reaction buffering step after the problematic chromogen incubation, which provided improvement in our case. And also the successful integration of HRP-based new chromogens into the sequential multiplex IHC procedure is also promising for the future of this technique. I would like to thank you for your interest and attendance on behalf of my team. And if you ever want to get in touch, you can just easily send an email to me or to Dr. Roll, and we will be very happy to communicate with you. Thank you.